Good afternoon. I'd like to thank the organizers for giving me the chance to present our work. This talk will touch on strategies to deliver nucleic acid cargoes to airway epithelia. These are my disclosures. So in this talk, I'm going to discuss gene addition approaches using integrating lentiviral vectors and gene editing approaches to restore CFTR function in airway epithelia. We have a variety of tools available for delivery. These include several viral vectors as outlined here. I'm going to focus on lentiviral vectors in this talk, and also non-viral strategies such as amphiphilic peptides and other non-viral delivery strategies. And I'll be discussing CRISPR ribonucleoprotein delivery. And importantly, we have animal models to evaluate how these work. So I wanted to briefly review some published work with lentiviral mediated gene delivery of CFTR expression cassettes, which we did in a CF pig model. And this was published in 2016. In this study, we used a FIV lentivirus pseudotyped with the GP64 envelope to deliver a CFTR expression cassette to the airways of newborn CF pigs. And after 2 weeks, we harvested tissues and performed several studies. I wanted to give you a highlight here where in freshly excised tracheal and bronchial tissues, we looked at CFTR-dependent anion channel activity. And compared to untreated controls in the trachea, we saw nice CFTR-dependent currents, and also in the bronchus. Similarly, with FIV CFTR, we saw partial restoration of CFTR channel function. So from these studies, we were able to show that we achieved in vivo correction of CFTR function using a lentiviral vector. And subsequently, we've been wondering if we could further improve the CFTR expression cassette. So one approach we're taking is codon optimization of CFTR. And our overall hypothesis is that transduced epithelial cells would express higher levels of CFTR, mRNA, protein, and anion current compared to wild-type CFTR. So this work is on a poster at the meeting number 517, presented by Laura Marquez-Loza, and you can look there for further information. So Laura is investigating 3 different constructs. These are schematically color-coded as shown here in panel A. And the 3 constructs are numbered 1, 2, and 3. Number 1 only codon optimizes the TMD1, while the others are codon optimized across the gene. And when you look at the GC3 content, the GC richness of the 3rd nucleotide of the codon, you see that CFTR3 is very enriched there compared to the other 2 constructs and wild type. The first thing that Laura did was clone these different constructs into pcDNA3 and then look at their expression profile in HEK cells. So when they're transfected in these cells, we see that all 3 constructs generate more protein than wild type CFTR, and that's enumerated here. She also looked at the mRNA abundance, and there was a trend that CFTR3 gave us more mRNA. These constructs were electroporated into FRT cells and their current properties studied. And in the presence of cyclic AMP agonists, CFTR number 3 gave us the greatest currents, and that's shown here in the bar graph on the right. So for additional studies in primary CF epithelia, the wild type and codon-optimized CFTR3 were cloned into HIV vectors with a weak constitutive promoter, as shown in the diagram here. And then basal cells from CF donors were transduced with MOIs of 0.1 to 1. And we studied the bioelectric properties of these cells when they were well differentiated in Using chambers. So in the left-hand panel here, using a GFP expression cassette, we show what percentage of cells are expressing the transgene when we transduce basal cells with these different MOIs. And with an MOI of 1, 75 to 80% of the cells are GFP positive. When these same cells were similarly transduced with the CFTR constructs, the short circuit current summary is shown on the right-hand panel. And at all MOIs tested, the codon-optimized CFTR3 construct gave us higher CFTR currents. So in summary, codon-optimizing CFTR yielded a cDNA that conferred greater cyclic AMP activated chloride current than wild-type CFTR when expressed in differentiated CF epithelia. And since airway epithelia are linked by gap junctions, enhanced expression of CFTR in a subset of CF epithelia may increase anion channel activity. And we plan to test this further in CF animal models. I'd like to discuss CRISPR ribonucleoprotein delivery now. And the first strategy we've used is delivery with amphiphilic peptides. This is done in collaboration with Feldon Therapeutics, and their technology uses peptides that have an N-terminal endosomal leakage domain joined by a linker to a C-terminal cell-penetrating peptide domain. And when these peptides are in solution with a variety of protein cargos, they drive the protein across the cell membrane rapidly, allow it to escape endosomal destruction where it can transit to the nucleus and do its business. And I'd like to just briefly show some data from a recently published paper where we evaluated this technology in an animal model. So these ROSA 26 MT-MG mice have an mCherry cassette and a GFP cassette. And the goal in this experiment is to deliver RNPs that will cut out the cherry, and you will see a red to green color change if this is affected. So we gave this material to mice intranasally and waited a week and analyzed the results. And what we saw was diffuse expression of GFP along the large airways and small airways. When we looked at this at higher power, we saw that we transduced ciliated and non-ciliated cells in the airway, also type 2 cells, but we did not see transduction of alveolar macrophages. And when we quantified the transduction efficiency or the editing efficiency, these are the results that we saw in the large and small airways. More recently, we've been working to see if we could further optimize these peptides and achieve higher degrees of editing. And this shows the optimization parameters we've been exploring with Feldon. New peptides have been generated. We've mixed these with Cas12a or Cas9 RNPs and investigated their editing efficiency in well-differentiated cultures of human airway epithelia. 3 days after transduction, we amplify the DNA and do next-gen sequencing. And there's a poster on this. Sumba Turari has this poster number 430. So this shows the sequencing results looking at the percentage of indels here. And you can see from our previous best peptide, we've made significant improvement in our delivery efficiency. And also for Cas9 RNPs, we've improved the editing efficiency and delivery. So to further analyze this, we've taken a subset of the best peptides and looked at their performance in the same animal model. And this shows results with one peptide delivering Cas9 RNPs. And you can see in the large airways diffuse editing with the green cells, and shown here in higher magnification. So we achieved editing in large and small airway epithelia with a novel peptide, and our current goal is to further study the best performing peptides. So this iterative design modification approach yielded peptides with superior delivery performance in airway epithelia compared to our previous best peptide. And from these studies, we also find that the optimal delivery peptide sequence depends on the cargo that we want to deliver. And some of the considerations are the size of the protein, and in the case of CRISPR nucleases, the length of the guide RNA. And preliminary in vivo testing of a few of these newer peptides shows that we can achieve editing in vivo. And we plan to continue to use these peptides and investigate CRISPR ribonucleoprotein delivery. And we're especially interested if this will work with base editors. So I'd like to also briefly talk about some early stage studies with adenine base editor protein delivery. So as we know, with class 1 CFTR mutations, there's an unmet need for new therapies. And according to the CFTR2 database, 46% of the single nucleotide mutations in CFTR could be corrected using an adenine base editor. So we've been collaborating with David Liu and Greg Newby at the Broad Institute to look at this concept. And we're focused on adenine base editor protein delivery. So in this model, there's an A residue, and the goal is to convert this to a G through an inosine intermediate using this technique. And so the workflow for the experiment that I'll describe involves using CUFI3 cells, which have the R553X mutation. So these cells were electroporated with the base editor and guide RNA, and then expanded in vitro. And some cells were used for next-gen sequencing, and some were seeded on transwells and allowed to mature so that we could do oosing analysis. So this shows the results of the sequencing analysis. So on the left, I show the R553X locus, and the A at position 5 in the editing window is our target for editing here in blue. And on the right, I show the next-gen sequencing results. So for the mock transfected cells, we see the expected 50% have the A at this allele. But after editing, this is converted to greater than 90% of the now wild type allele. So the majority of mutant alleles had successful conversion of an AT pair to a GC pair. And we saw no significant incidence of bystander edits or indels. We also have recently had the opportunity to look at the functional correction of these cells. And so on the left, I show short circuit current tracing data from R533X cells. So in red are the mock cells, in blue is a non-CF control, and in green are cells that were edited with the ABE. And on right, I have summary data for multiple epithelia that were studied. So in green, compared to the values on the left here in the mock transfected, you see that we've restored significant CFTR chloride current, and in blue is a non-CF control. So the ABE reversion of the R553X allele was sufficient to restore CFTR anion channel activity. So in summary here, molecular correction of R553X mutation was achieved using an ABE protein. And the base editing efficiency for this mutation was high with negligible bystander edits or indels. And functional correction was achieved in CUFI3 cells. So in the future, we would like to further optimize ABE delivery, and we're using both viral and non-viral approaches to achieve this. And we'd like to know if we can correct the CFTR gene in airway progenitor cell types. And with that information in hand, we'd like to test these tools in human primary epithelia and in a CF animal model. So I'd like to acknowledge folks in my lab who worked on this project, especially Satish Krishnamurthy and Sumber Terori and Laura Marquez Loza, doing the codon optimization work. My longtime collaborator, Pat Sin, and Ashley and Christian, are collaborators at the Broad Institute, collaborators at CHOP on the Lenti project, my colleagues at the University of Iowa, and David Gee and collaborators at Felden Therapeutics and our funding support. And I'd like to thank you, and I'd be happy to take questions.
My name is Mark Salzman. I'm delighted to be able to present in this symposium today. I'm going to be talking about design of degradable polymer nanoparticles for the delivery of gene editing agents to cystic fibrosis. This work is a collaboration with my colleagues Peter Glazer and Marie Egan at Yale. Also, lots of students and postdocs have been involved with these projects, and I'm going to try to mention them and their contributions as I go. First, a brief disclosure. I have the following relationships with NanoSiv and Stratify. NanoSiv is focused on developing biodegradable nanoparticle sunscreens, Stratify on nanoparticles for local delivery of cancer, neither of which are topics I will refer to today. What I'm going to talk about is using synthetic biodegradable polymers like PLGA, which is shown here, that are loaded with agents to accomplish gene editing. And these particles, we've elected to work on these particles for gene editing because of some interesting observations that have been made using these particles to deliver drugs. One, that the particles themselves are not toxic if you add them even at large concentration in cell culture. You can load compounds with drugs like this chemotherapy drug here, and they're released slowly over time, sometimes over a period of multiple weeks. If you add those drug-loaded particles in culture, they can be very effective at delivering the active ingredient, the agent, as shown with these chemotherapy loaded particles that are able to kill tumor cells in culture. And when injected in animals, these particles often have a pharmacologic effect, like the one shown here. When the particles directly injected into a tumor, they arrest the growth of the tumor. And these last two qualities of nanoparticles seem to be related to the fact that the particles themselves are much smaller than cells. And so the particles can enter cells, and you see that in this rotating confocal micrograph, where you see the red outline of a cell, you see the nucleus in blue, and you see green nanoparticles that have, within a short period of time after being exposed to the nanoparticles, the particles appear inside the cell, and they can release the active ingredient that is loaded into the particle, and they release it intracellularly. So these particles can be used as carriers for drugs. What I'm going to talk about today is shown in outline form here. I'm going to talk about using this kind of nanoparticle technology to deliver gene editing reagents that were developed by Peter Glaser in his laboratory. These are based on peptide nucleic acids, and I'll describe that in just a moment. I'm going to talk about converting them into an inhalational nanoparticle delivery system that can accomplish gene editing in the CF lung. Talk about using them on intravenous delivery in animals in utero, and also for intravenous delivery for multi-organ gene editing in mice. And then talk about a bit about the future, where I think we could go for engineering of more efficient delivery systems for these kinds of materials. First, nanoparticle and peptide nucleic acid gene editing technology. So this is work that was pioneered by Peter Glaser, and the idea was to identify a stretch of genomic DNA where you want to make a change, and so there's a mutation somewhere in the genome, and you design a donor DNA template that's nearly totally complementary to that stretch, except for the nucleotides that you would like to change. Those are different in the donor DNA. And then you design a peptide nucleic acid, or PNA. These are nucleic acid analogs that have a peptide backbone, but bases that occur with irregular spacing, like in nucleic acids. And you design this to bind at a region near the site that you want to repair. When that binding occurs, a triple helix is formed at that local site, which instigates the cell's own machinery to repair that triple helical defect. And when it does that repair, on occasion, it can incorporate the template that you have provided, and so create a new strand of DNA that has your sequence built into it. And in the early days, we used, Peter's lab used allele-specific PCR in order to identify whether you'd made the correct change or not. Now we use other PCR-based techniques, and we look for phenotypes that are associated with this change in gene sequence. Well, this is early work, and it was done by an MD PhD student named Nicole McNear, where she loaded this PNA and template DNA into a polymeric vector. In this case, it was PLGA, polylactide glycolic acid, and then injected, either applied to cells in culture or injected intravenously into mice. And what she found in cultured cells, and these were hematopoietic stem cells, was that if you put both the PNA and the DNA inside the polymer vector, that you could accomplish fairly efficient gene editing as detected by allele-specific PCR. She also found that if you injected these particles directly into animals, you could find gene editing in tissues throughout the animal, including in the bone marrow, in the lung, in the liver, in the spleen, in a variety of different organs. And that was also measured by this allele-specific PCR. So how could you use that for inhalational delivery for gene editing in the setting of cystic fibrosis? Well, why use nanoparticles in this setting? One is that it's well known that if you breathe in nanoparticles, if humans breathe in nanoparticles or animals, they get widely distributed throughout the lung, and particles that are about 100 nanometers or so in size are ideal for distributing throughout all the lung space. In addition, it's known that nanoparticles that have the right properties can penetrate through the mucus barrier on the epithelial surface of the airways and the alveoli, and that's been shown by work like this from Justin Haynes' group, where pegylated particles of the right size can migrate efficiently through mucus. So we used that understanding to design nanoparticles that we thought would be efficient at delivering these reagents to the lung epithelium. These were largely made of polylactide coglycolide, but we also added the cationic polymer, PBAE, into the mix so that it would more efficiently load nucleic acids because of the electrostatic interactions between the cationic polymer and the anionic nucleic acids. We put polyethylene glycol on the surface using a lipid to facilitate its accumulation on the surface, and we attached a cell-penetrating peptide to the surface in order to enhance uptake by epithelial cells. And you can see in vitro, with these changes made in the particle, we could get very robust accumulation inside epithelial cells. We gave these intranasally to mice. We dissociated the lung and looked by flow cytometry, and just one result here showing that with all these modifications made to these nanoparticles, almost all of the epithelial cells were exposed, contained, were associated with nanoparticles after a single inhalation. We use this to treat cystic fibrosis in the animals by loading the nanoparticles with a triplex forming PNA that binds downstream of the F508del mutation site, and we also delivered a donor DNA with the correct sequence in order to serve as a template. In human cystic fibrosis bronchial epithelial cell cultures, we identified the mutation in clones. And if you cloned out those cells and then grew them in culture and measured chloride flux, we found that they were almost as efficient at moving chloride as the normal cells without the delta 508 mutation. We deliver these intranasally in animals and then measured the nasal potential difference after some weeks after delivery. And this is work that was done, all this work was done with Marie Egan, who many of you know, and saw that there was a statistically significant change in NPD that brought it much closer to the wild type condition. And if you looked inside the, if you collected cells and then measured percent correction, and we did this by deep sequencing, we found that with these nanoparticles, we got in culture cells almost 10% gene editing, and in the living animal, 1 to 6% gene editing at different, with cells collected at different parts of the respiratory system. This has been published, and the citation is here. Well, we wanted to test whether this successful gene editing could be extended in a variety of different ways, particularly if you could do it by intravenous injection instead of by inhalation, because more than one organ is affected by, although the lung is very much affected, it's not the only organ that's affected by cystic fibrosis. And our first test of this was done with David Stittleman, who's a pediatric surgeon and an expert in fetal treatments. And he developed a method for injecting these nanoparticles into the circulation of a mouse in utero. And these are fluorescent nanoparticles, which you could see this micro needle is going into the vitelline vein, and when the nanoparticles are injected, they fill the circulatory system of the fetus. Those particles stay with the fetus, and you can measure them by IVUS imaging, as I shown here. These are particles that are introduced at different times, either by intravenous injection, as shown above, or by intraamniotic injection, which is not shown here. But after the pups are born, you see that the mother is left with no fluorescence. So all the fluorescence goes into the fetus, and none of it is detected in the mother. If you look inside a fetus, we find that with IV injection at E15.5, most of the particles go to the liver. At later time points, with intravenous and intraamniotic injection, we find a lot of particles also go to the lung. So based on this, we decided to test the possibility of in utero gene editing in the delta 508 mouse. And the idea here was to inject nanoparticles intravenously at gestational day E15.5, as shown here, or to inject them interamniotically at gestational day 16.5, which is after the fetus has started breathing the amniotic fluid. And then we would test these, the mice after they were born for nasal potential differences, rectal potential differences, and other functional attributes. I'll just show you one result from that now, and this is the PhD thesis work of Adele Ricciardi. So untreated delta 508 mice with their abnormal nasal potential difference, if they're treated interamniotically, there's an improvement, but it's not dramatic. More dramatic with intravenous treatment down into the normal range and comparable to wild type mice. And this was measured 4 months after the animals were born. When Adele looked for gene editing by digital droplet PCR, she found that there was appreciable editing, 1 to 2% in the liver, the lung, the kidney, the heart, even in the brain, and more variable, but sometimes higher levels of editing in other organs that would be of interest to the CF community, including the pancreas. Well, this was our first demonstration that you could do gene editing in a living animal by intravenous injection, but we wanted to see if this would work in adult animals as well. And this is the work of Alexandra Piotrowski-Daspit. I'm going to just brush over it because it's described in her poster, which is shown here. Here we've used PNAs that were enhanced by a modification in the gamma position of some of the monomers, so that it had enhanced binding and enhanced gene editing activity, and that's described very clearly in her poster. She has tested these again with Marie Egan, both in cell cultures and cells that are cultured in an air-liquid interface and measuring short circuit currents. You can see a pretty significant change in short circuit current with these improved nanoparticles, and you see fairly high levels of gene editing, up to 30% in this case here, with 3 treatments in vitro. This translates into success in treating animals. These are animals that were intravenously injected with multiple doses of these nanoparticles, 4 doses over a couple of weeks. And in a significant fraction of the animals, you see changes in the nasal potential difference, also changes in the rectal potential difference, suggesting that you've had phenotypic impact in both the respiratory system and the gut. Lastly, I want to talk about the future and how could you engineer more efficient polymer delivery systems. And I'm going to talk about 2 approaches that we have been testing. One is a completely different polymer that we've synthesized on our own. It's polyamine coester. It's a cationic polymer, so you form it from a set of monomers, and you can adjust its composition. We've described this in papers, which I'd be happy to share with you. Because of its cationic nature, it associates with nucleic acid to form either polyplexes or solid nanoparticles, depending on the hydrophobicity and molecular weight of the polymer that you make. But we find that these at least have the potential to be even more efficient carriers of these nucleic acid gene editing reagents. And the second category I'm going to just hint about are block copolymers of polylactic acid and hyperbranch polyglycerol. And this is hyperbranch polyglycerol here. It's, in this case, substituting for polyethylene glycol. You can see it's a branched molecule instead of a linear one. And we think that that's going to give it better properties in terms of mucus penetration, and also has some unique characteristics which allow us to both load agents more efficiently and to deliver them more efficiently. First, for the PACE or polyamine coester system, and just why we're so excited about it, shows a comparison in vitro between the PLGA systems I've been describing before, and some of these PACE systems, where even at a lower concentration, we find substantially greater gene editing in vitro. And we also know that we can change the chemistry of this PACE nanoparticle by changing the composition of the macromolecules that go into making the nanoparticle. And in some of those compositions, we see more dramatic accumulation in the lung than we see with PLGA. And so that might also be useful in enhancing gene editing in this situation. One demonstration, a further demonstration of the potential value is that we've used PACE to deliver messenger RNA directly to the lungs by inhalation in mice, and you can see we delivered here mRNA for luciferase, and by IVUS imaging you see high levels of expression of luciferase in the lungs of these mice. If we measure that quantitatively, you can see compared to a commercial transfection reagent, we do orders of magnitude better, and that we can optimize the delivery of mRNA by subtly changing the composition of the nanoparticle. For PLA-HPG nanoparticles provides several potential advantages. One is that if you inject it intravenously, it circulates for a much longer time than PLGA alone. Here shows a half-life of almost 10 hours of circulation. And so this would allow it to circulate long time in the body, reach the lungs, and potentially other organs as well. If you deliver it intratracheally by inhalation, you see none of it becomes circulating, but it all ends up in the lung. And so you could really focus activity in the lung by intratracheal or intranasal delivery of particles like these. The other reason we like it is that we've shown that it greatly improves gene editing in vitro. This is PLGA, which gives us in these cultured cells gene correction that's in the 5 to 10% region with a single treatment. But some compositions of these PLA HPG can reach up to 20% in one single treatment. And we think that offers us a potential advantage in improving the therapy. I should say that this last work here was done by Lexi Subari, Molly Grun, and Elias Quijano, who are all current members of the laboratory. So in summary, polymer nanoparticles composed of PLGA and triplex oligonucleotide gene editing ranges can produce phenotypic changes in CF cells and in the tissues of CF animals after in vivo in situ treatment. Engineering of these polymer nanoparticle properties allows for effective delivery in animals by either inhalation or by intravenous injection, and new polymer compositions such as the PACE and the PLA-HPG, which I've described, may enable more efficient gene editing. I mean, just stop by first acknowledging funding from the Cystic Fibrosis Foundation, grateful for their support and all the helpful conversations, and from the National Institutes of Health. Thank again Peter Glazer and Marie Egan, who have been fabulous collaborators and are also fabulous people, and so it's a pleasure to work with them. And the postdocs and graduate students I mentioned, Nicole McNear, Elias Quijano, Rachel Fields, Adele Ricciardi, Alex Piotrowski-Daspit, Lexi Suberi, Molly Grun, and Laura Bracaglia. Thanks for your attention.
Hello everybody, I'm John Lewick, and today I'll be talking about our research on suppressor tRNAs and their potential use as therapeutics for nonsense-associated cystic fibrosis. I'd like to start off by thanking the organizers for giving us the opportunity to talk about our research today. And we have no disclosures that need to be made. So 10 to 15% of all genetic diseases are caused by nonsense mutations, also referred to as premature termination codons or PTCs. PTCs affect about 3 million people in the United States alone and are the cause of about 7,000 different diseases, including cancer. So here I have a schematic of the normal translation process where the ribosome binds the mRNA and acts as a decoding center where transfer RNAs translate the mRNA code into protein. So this process occurs until it hits a stop codon, and the stop codon acts as a signal for the dissociation of this ribosome from the mRNA. And in normal circumstances, the result is a normal full-length functional protein. However, in the presence of a premature termination codon, there is a premature signal for the ribosome to dissociate at the PTC, and the result is a truncated non-functional protein. Additionally, the PTC signals for the degradation of the mRNA through processes called nonsense-mediated decay. So really, nonsense-associated diseases are a two-hit disease, one in which you have a decrease in total functional protein, and two, a decrease in the mRNA expression. And so now, without protein, the therapeutic target must shift to either the genome, where techniques like CRISPR can be used to fix the genetic misstep. You can target the translation processes, and several groups have been successful in finding small molecules that bind to the ribosome and promote read-through of PTCs and full-length protein. Another is targeting the nonsense-mediated decay pathway, and the result of this would be an increase in the steady-state expression of the mRNA transcript. The approach that we're taking is the use of suppressor tRNAs, and we call these anti-codon engineered tRNAs or ACE tRNAs. So on the left here, I just have a schematic of how normal tRNAs participate in translation. So you have a tRNA that is recognized by the synthetase, and this interaction is keyed. And so this tRNA is charged with the canonical amino acid by the synthetase. This acylated tRNA is then recognized by a protein called EF1 alpha, which protects it from becoming deacylated and also directs it to the ribosome where it can participate in translation. So on the right is our ACE-tRNA approach, where we've engineered the anticodon to no longer recognize the cognate codon, but now recognize one of 3 stop codons. And we know that when we do that, that the synthetase still recognizes the ACE-tRNA and charges it with the correct amino acid, that the acylated tRNA is still recognized by the EF1 alpha, which directs it towards the ribosome, where now it is able to suppress PTCs and generate full-length functional protein. So one of the benefits of using the ACE-tRNA approach is that not only do you get full-length protein, but you get full-length wild-type protein, and that's because these ACE-tRNAs suppress with the correct amino acid. So we performed a screen, which we published last year, where we looked at over 500 ACE-tRNA sequences, and we developed a high-throughput cloning and screening strategy using nanoluciferase with a premature termination codon in it. And so when you suppress that PTC, you get luminescence. And so we looked at PTC activity of our suppressor tRNAs with ACE-tRNAs promoting high levels of suppression having more luminescence. And so here in this table, you can see that we have sequences of very efficient ACE-tRNAs for several amino acids. And the reason that these amino acids were picked because their codons were 1 nucleotide away from a stop codon. So now that we have this a large library of ACE-tRNAs, obviously the next huge step is to deliver them to the lung. So here we have a schematic of the tRNA gene, and one of the attractive attributes of tRNAs is their compact size. So the entire expression cassette is shown here. So it is about maximally 90 base pairs in length, and that's because the promoter for tRNAs is intragenic. And so the necessary sequence for expression is within the coding region, with some few exceptions, which we'll talk about later. So because of its compact size, you can encode them in viruses, you can encode them in DNA, and also you can deliver ACE-tRNAs as RNA. And obviously this would come with encapsulating them in chemistries like nanoparticles. And so my lab is working on all 3 of these approaches. And we realized quickly that in order to determine how efficient these approaches are, we needed to come out with a PTC reporter assay. And so here we've generated a piggyback-based PTC reporter construct, and it will integrate a nanoluciferase with a PTC. And the beauty of using these piggyback transposon approach is that the integrations are a single event into the genome, and which results in very stable genomic landscape, which sometimes isn't offered by standard approaches for making stable cell lines. We've made three formats of nanoluciferase PTC reporter. This is just the nanoluciferase with the PTC. And then we have one with a pest destabilization sequence. And so the half-life of this reporter is about 20 minutes. And then we have a secreted format where we can go in and sample the media to look for PTC suppression efficiency in a monolayer of cells that is not disturbed. And we can look at this longitudinally. So what we've done here, is shown in these graphs, is we've taken a couple of our winning ACE-tRNA sequences that are very efficient at suppressing PTCs, and we've shown that no matter the format, that you still get a sufficient signal, and that the shape of the efficiency isn't affected by the format of the nanoluciferase, meaning that the readout isn't affecting what you would expect in the PTC suppression efficiency. So the first thing that we did was we made a 16 HBE cell line that has the R1162X mutation in the CFTR gene with the secreted format of our nanoluciferase. And then we transfected those cells with plasmids that encode the arginine ACE-tRNA. And we sampled on day 1, 2, 3, and 4, and this was done in trans wells. And this is just taking the apical media and putting it in a plate reader. And you can see that you get robust PTC suppression. You have a huge signal here, but to our surprise, it decayed quite rapidly over 4 days. And so this also occurred with our ACE tRNAs that encode the leucine. So this has nothing to do with the arginine ACE tRNA, but has to do with it being encoded as cDNA. And so this actually was quite a good explanation as to why we were getting really weak rescue in our leucine chamber experiments, which were measured out at day 6 and 7. And that's because all of the PTC suppression activity had gone away by that time. So our explanation for this decrease in PTC suppression activity is that we have silencing of our plasmids because of the bacterial sequences that are encoded in these plasmids. And also there's a dilution of the plasmids because of cell division. So 16-HBE cells split quite rapidly. And when they form monolayers, they still seem to continue to split. And so we're dealing with the silencing of the plasmids by removing some bacterial sequence in them to see if we can prolong the activity of the ACE tRNA. And there's little we can do about the splitting of 16-HBE cells. But of note, in vivo, the cells that we deliver our technologies to in the lung won't be splitting rapidly, and so this shouldn't be a problem therapeutically. Next, we wanted to see what the half-life of our ACE tRNAs are as delivered by RNA. And in order to do that, our reporter had to have a shorter half-life than that of the thing that we were delivering. So we went to our PEST nanoluciferase, which again has a half-life of 20 minutes. And we made stable cell lines in 16-HBE GE cells that again have the R1162X mutation in CFTR. And we delivered RNA that was the arginine ACE tRNA. And so we really didn't know what time frame to look at here. And so we just started really early and went on to 30 hours. But what you can see here is that the RNA was active very early. And within 2 hours, we were seeing PTC suppression of our reporter, which peaked at about 4 hours and then decayed rapidly down over until 30 hours. And when we fit this decay, we show that we get a half-life of the RNA to be about 6.5 hours. And so for therapeutic use, this tells us that the RNA may need to be modified in a way that increases its stability or formulated and delivered on a daily basis to maintain levels within the therapeutic window. And so we were unhappy with the fact of not being able to measure PTC suppression and rescue of CFTR protein function. And so we developed a piggyback system where we can incorporate several copies of ACE tRNAs stably. And we call this our PB donkey system. And this is based off of the P mule system developed by the Fru lab, where they use the same recombinase-based cloning, but instead they generated lentiviruses. And so using this approach, you have what's called donor plasmids, which you can encode any gene of interest into. What we did is we put 4 copies of our ACE tRNAs into each one of our donor plasmids and then recombine them into the transposon, which then can be transfected into cells with the transposase for stable integration. And the reason that we took this route is because when you're dealing with repeat sequences of DNA, they become very stable. So we can make our manipulations in rather small plasmids with that are very stable, generate or kind of large unstable plasmid that is the transposon, but no longer have to make manipulations to the sequence. And so when we did that, we were able to make stable 16-HBE GE cells with the R1162X mutation with about 16 copies of our tRNA. And when we did that, the cells were healthy, they split normally, and they were able to form tight junctions. And so you can see here is a representative trace of just wild type 16-HBE cells. And then in blue here are cells that express 16 copies of our arginine ACE tRNA. We also made cell lines that stably express just 8 copies of our tRNA. And so what you can see here is that with 8 copies, you get about 2% rescue in CFTR function. And with 16 copies, you see that you get about a 7% rescue in CFTR function. And so what this tells us is that we're probably not saturating this effect, and that if we can get more copies of arginine ACE tRNAs into the cell, that the rescue of CFTR should be greater. And importantly, we expect to be developing delivery strategies that will far exceed only 16 copies of arginine ACE tRNAs. And so moving on, we look to see if these ACE tRNAs can suppress NMD. So here we have just the exposure of our R1162X and W1282X 16-HBE cells with G418. And you can see that you have a significant rescue in the CFTR expression, however, it is modest. When we go to our plasmids, you can see that after 48 hours, with 1 in 4 copies encoded in our plasmid, that you get a robust rescue of the CFTR mRNA expression that is near 40–45%. And then when we transfect in our leucine ACE tRNAs into W1282X cells, that we get even a larger rescue, exceeding that of 50%. And of note, our use of the leucine tRNAs, because they have a higher activity, shows, that is better than that of the tryptophan ACE tRNA, which we were originally using. And so the ability for us to pick and choose ACE tRNAs and encode different amino acids really just displays the kind of a la carte, I guess, a promise of our ACE tRNA library. And the next thing we wanted to do was to see if we can deliver our ACE tRNAs as RNA and also rescue NMD. And so looking at just in 6 hours, delivery of our arginine as ACE tRNA as RNA rescued to about 30% that of wild type. However, this diminished after 24 hours. And so again, looking back at delivery, we will be delivering our ACE tRNAs to the lung as RNA, DNA, and virus. And we had the thought that if we can increase the potency of our ACE tRNAs, we wouldn't have to have such a high delivery efficiency. And so if you look at the tRNA gene, which is shown here, there are some upstream control elements that are important for transcription. They're not necessary, but they can enhance transcription. And then you have a trailer sequence that is important for processing of the tRNA. So kind of shown here is the tRNA gene with transcription elements bound to the A and B box and the upstream control elements. Once you have transcription, these 5' leader is cleaved off with an enzyme called RNase P, and the 3' is cleaved with RNase Z. And then this tRNA is further processed and modified where it participates in translation. So our thought here is that we can screen about the endogenous upstream sequences. And so again, there are 415 tRNA genes, and there are 386 unique UCE elements. And so we wanted to screen all of these to see if we can increase transcription and then obviously increase the ACE tRNA suppression efficiency. And there are 256 different combinations if you look at 4 base pair sequences of trailer sequence. And so what Joe Porter did in the lab is he generated a high throughput cloning and screening plasmid in which you can reference the rescue of nanoluciferase with a PTC to that of FLuc of firefly luciferase. And so you can normalize your data that way. And we looked at 386 sequences of 55 base pairs of upstream control elements. And the data is shown here in this heat map. And the line here is the original sequence. The next thing we did is we looked at 256 sequences in the 3', and here's the heat map of our results. And you can see that we had several that were higher than the original. If you took these sequences and we took the top 12 of each and we transfected these reporter plasmids into HBE cells, you see that in many of these sequences give over 100% enhancement of PTC suppression, and that one of these 3' trailer sequences actually increased PTC suppression by 65%. And so our goal with that is to combine those. Because they use two different pathways in tRNA processing, we think that they could be additive. So in summary, we've generated a straightforward piggyback PTC suppression system that is not confined to a specific cell line. We can actually transfer it to other cell lines as they become developed. We've shown that ACE-tRNAs suppress endogenous PTCs in the CFTR gene. ACE-tRNAs are effective at increasing the steady state levels of CFTR mRNA when encoded as DNA or delivered as RNA. And this is likely through promoting the completion of the pioneer round. And ACE-tRNA ARG suppression efficiency can be enhanced through optimization of the extragenic 5' and 3' sequences. And so next, we're going to put this all together and tackle the delivery hurdle. And so if you're interested in what I talked about here, go and visit poster 387, where the graduate student Wuri has what I think are beautiful results, and which were talked about in this result, and they're shown here. And postdoctoral Porter, who performed a high throughput screen, and that poster is number 460. And these are the members of the lab. And lastly, I'd like to thank the CFFT for providing us with the 16 HBE-GE cells and our collaboration with IDT, and of course the funding that's come from through Vertex, the Cystic Fibrosis Foundation, Emily's Entourage, and U of R Ventures. And thank you very much.
It's a real pleasure to be part of the 2020 North American Cystic Fibrosis Conference. My name is Richard Worcester, and I'm the Chief Scientific Officer of Translate Bio. And I'll be talking about delivering CFTR mRNA, an inhaled, mutation-agnostic approach to treat cystic fibrosis. For my disclosures, I'm an employee and shareholder of Translate Bio. And the overview of my talk, I'll describe the continuing unmet need in cystic fibrosis and how we have discovered mRNA therapeutics, applying that to MRT-5005, the first CFTR mRNA therapeutic being evaluated in individuals with cystic fibrosis. But beyond that, I'll also describe our next generation CFTR mRNA. So to begin, in this timeline here, you can see how this really large improvement in life expectancy, predicted survival, from the days when CF was first described over the time to current days. In parallel to that, we can see the discovery of DNA, RNA, and that ultimately leading, I think, to the seminal component where the CFTR gene was identified. And here almost as a branch to then moving to the small molecules, the modulators, which have gained approval and have had a significant clinical impact. But then in parallel to that, how you could actually address the gene directly, whether through gene therapy or through using an mRNA, such as with MRT-5005. And even with the benefit that we see with the modulators, there's still an unmet need in patients with cystic fibrosis. Now, this data here, taken from the registries from the US, from Europe, from Canada, and Australia, you can see that a large proportion of patients are eligible for either ivacaftor or the elixacaftor triple combination. There remains almost 10% of individuals whose genotype suggests they're not amenable to these modulators. But the story is more complex than this, because when you look at the clinical data for the triple combination, there's a proportion of patients who see no FEV1 benefit, and others who have a low level of FEV1 response. And so this unmet need expands from around that 10% mark to almost 30% of patients with cystic fibrosis. Now, an mRNA therapeutic has the potential to restore CFTR expression in a mutation-agnostic approach to help that 10% who are not amenable, but possibly all patients with cystic fibrosis. Now, our approach to using mRNA to do this is described here. And so we start in the top left-hand side of this figure with this individual. They're inhaling an mRNA which is encapsulated in a protective lipid nanoparticle. When this enters the lungs, it then is taken up by cells, and you can see following the arrows how as the LNP degrades, the mRNA is released from that lipid nanoparticle. And from this point, the cellular machinery takes over, essentially translating the CFTR mRNA and creating protein, and that protein being transported to the cell surface as would happen in a healthy lung cell. As you can see, we've also included the genome here because mRNA is not the only way you could achieve the expression of an intact normal CFTR protein. There are these other genetic modalities. However, our mRNA approach is distinct from both, say, gene editing and gene therapy, which are shown here. The mRNA doesn't integrate into the genome of the patient, and so it doesn't permanently alter the genome. And we know that altering genomes can have unexpected off-target effects, and which are not desirable. In addition, we can titrate the dose. So we can either increase, we can decrease the dose of the mRNA to optimize it for benefit. And we can change, for example, the dosing frequency, all with these pharmaceutical properties that you want to really tailor the treatment so it has maximum benefit for patients while looking to minimize the off-target effects. And not only that, we know that mRNA can be given in a repeated dosing fashion for chronic diseases such as cystic fibrosis. Now, beyond CF, we believe mRNA has advantages in many other types of genetic diseases, and possibly even beyond genetic diseases, where you could expand into disorders where introducing or boosting the level of the desired protein could have a therapeutic benefit. Now, to achieve this vision of having an mRNA therapeutic, we need the components of being able to manufacture the mRNA and then deliver it to the cells of interest. So first, manufacturing. The mRNA that we synthesize contains the features that you would find in a natural mRNA. So if you look on the left-hand side of this slide, you'll see we have a 5' cap. We put the poly-A tail to protect the RNA. The 5', the 3' untranslated regions that flank, in this case, the coding sequence for CFTR. As we make that RNA in the center panel, in this analysis, you can see that large high peak, that's the CFTR mRNA. And so we're looking for purity here, as well as quantity that we can produce. And the reason for looking for that purity is we want to be producing as much full-length CFTR protein as possible. If we have truncated RNA, it could lead to truncated protein, which is just then not effective, possibly could even be toxic. So really important to have that purity as well as the quantity. And to demonstrate that this RNA can produce protein, on the far right-hand side, you can see specifically looking at CFTR protein production. It's the molecular weight that we would expect, and it's reproducible from one batch to another. And later in my talk, I'll show how this protein is functionally active as well. So having made the RNA, next we package this into our proprietary lipid nanoparticles. And there's a lot of detail on this slide, and I'll walk you through again from left to right. So our proprietary lipid nanoparticles, these contain the mRNA and ionizable lipids, helper lipids, and PEG. And we formulate these to generate these lipid nanoparticles. They're around 100 nanometers in size, and these are what we can then deliver to the lungs. Now, in the actual data here, moving from the low resolution to high resolution, and really specific reasons why we do each of these. In the low resolution setting, this is really has a strong part to play in rapid screening of novel lipids, because we can administer this, in this case, the firefly luciferase RNA to these animals, and you can see the blue, the sort of green and red expression that we can detect in the lungs 24 hours after the RNA was administered. So this gives us a rapid and sort of more global aspect to protein expression, but doesn't give us resolution. In the center panel, we're getting more detail into the organ resolution of expression. These particular mice, they have a reporter gene, a tomato reporter gene inserted in their genome, but it's inactive. This can be activated by giving the mice a Cre-recombinase RNA, and this then acts with the lock sites to cause the gene to be switched on and the tomato reporter to be expressed. Now, there is some leakage. You can see in the saline control, there is some expression. And to orientate you there, the head of the mouse is at the top of the video, and the diaphragm is at the bottom of this screen. But when we add the Cre-recombinase mRNA to the LNP, if you look in the center there, you can see the trachea is fluorescing, is lighting up with the tomato, as well as the upper branches to the airway. And this is because the Cre was taken up and expressed. And so this gives us a global aspect at an organ level. On the far right, getting to then specific cell-based expression using immunohistochemistry with an antibody to our protein of interest. And then through these sections, we can detect the level of protein expression, the location, which cells, where in the cells this is located, as well as things like a duration of expression of the protein. So using these components of our platform then led us to MRT 5005. And this is currently being evaluated in a phase 1/2 clinical studies called Restore-CF. So MRT 5005, what this consists of is an mRNA for CFTR packaged in a lipid nanoparticle, and it's administered to patients once a week through a nebulizer. Here you can see the design objectives. It's a safety tolerability study, eligibility criteria in terms of being over 18 with a range of ppFEV1 or FEV1 values here, and broad demographics in terms of mutation types and exclusion criteria. In part A, each patient received a single dose of MRT 5005. So there were 4 patients in each of these groups. 3 received 5005 and 1 received a placebo. The dosing in the multiple ascending groups is ongoing and is not part of this presentation. And the results from this single ascending group shown here for the 8, 16, and 24 milligrams in part A were presented at NACFC last year. I think it's important just to summarize the results that we presented. In terms of FEV1 changes, so we observed a marked increase in FEV1, mainly at the 16 milligram mid-dose level, as shown by the blue line in the graph on the left-hand side of the slide. And the individual FEV1 increases at 16 milligrams range from 11 to 22%. At the time of onset of those increases is consistent with data on the rate of CFTR expression in model systems. And I should note that the increases were higher than we were expecting, particularly if you then compare those with the known variability seen in FEV1 in CF patients. Of course, tolerability is a key component of this study, and at the mid and low doses, MRT 5005 was generally well tolerated. There were no serious adverse events at any of the dose levels. And in terms of adverse events, the most common were cough and headaches. There were transient febrile reactions observed, mainly in the high dose group, with symptoms occurring 4 to 10 hours after dosing, resolving within 24 hours, and patients leaving the study center as planned on day 2. So generally, 5005 was well tolerated at low and mid-dose levels. So in summary, we're encouraged by the results from the Restore-CF study. It's the first data from an inhaled mRNA designed to treat a chronic disease such as cystic fibrosis. And it provides information on the potential of mRNA therapies, not just for CF, but actually broadly with respect to pulmonary diseases. And the increase in FEV1, the safety tolerability profile were very encouraging to us. Following a pause in the clinical study due to COVID-19, we resumed enrollment and dosing for MRT 5005 in September. Now, in addition to MRT 5005, we're developing a next generation CFTR mRNA medicine. And this builds on our learnings from 5005, incorporates advances we've made in our platform, for example, including novel lipids, optimizing RNA sequences. And so really feel we have a number of gains that we can add on top of 5005. And in addition, we've been interrogating the biology of CFTR, because we feel that there's possibility there that we could make small, rational engineering changes into the CFTR protein that would actually improve the properties that we want to see in our target profile of at least matching, but hopefully increasing the clinical benefit, increasing tolerability, and reducing the administration time. There are three components that I will describe to you that we've evaluated through our work on our next-gen program. One is further optimizing the RNA construct itself. Second, I've already touched on, engineering the protein sequence. And the third are novel, more potent lipids that we have been identifying. Now, starting with codon optimization. So because of codon redundancy and what we know about this, we have been changing the codon usage, in this case, in the wild-type CFTR protein sequence. So while the RNA sequence itself is subtly altered, the protein is still completely functional, normal wild-type CFTR. Now, this can take advantage of tRNA availability, aspects of ribosomal pausing within the translation of RNA, and the consequence of these optimizations is shown on the right-hand side here. So this is how we've taken one of our mRNAs, and here we're looking at the functional outcome from that protein. This is an Using chamber analysis of chloride flow across a membrane of FRT cells. And you can see the blue line, that's our previous codon sequence and the level of current that we can achieve through that. But we see this increase in current with, in this case, showing 2 of our new codon optimized mRNAs. So this could mean either delivering less RNA to achieve the same extent of chloride flow, or possibly getting more chloride flow for the same amount of RNA. So I mentioned not only have we've been looking at the wild-type CFTR protein, but we've also been investigating rational changes, protein engineering, with the goal of increasing chloride flow. Now, this is one example here. I would encourage you to go and see poster 515 for further details on our work in this area. So CFTR, the protein, is regulated by, in some cases, phosphorylation at multiple sites in the R domain. Now, what we know from general protein biology is that the serines, the threonines that are phosphorylated in proteins, we can mimic that phosphorylation, and so almost have this permanently on phosphorylated state by changing those serines, threonines for example, aspartic acid. And this could lower the threshold for opening the CFTR channel and perhaps allow it to be open for a longer period of time, more chloride flow. On the bottom left-hand side, here we've plotted that as we increase the number of serines, threonines that we change to aspartic acid, you can see the phospholine EC50 decreases. So we're lowering that activation potential. And on the right-hand side, this is 2 examples for where we have both 13 or 15 aspartic acids compared to the wild type. And how, as you increase that phospholine concentration, you can see wild type, it takes a larger concentration of phospholine to have that activation compared to the particular constructs where we have the phosphomimetics. And finally, what I'll touch on is our lipid discovery activities. and where we've been able to significantly increase, in this case, looking at the expression of our reporter, our firefly luciferase reporter. So if you focus on the graph on the right-hand side, you can see we've identified a series of different lipid families where we've made small changes to the lipids in each of those, but they still fall within these families, and how those minor modifications can have a large increase in what we're calling the potency of the lipids here. And that potency, again, is where we give the same amount of mRNA but see this very dramatic increase in expression. And so now we've been incorporating this into our CFTR work. And this example here, in the top line on the left, you can see with saline, the DAPI is the nuclei of cells, but there's no CFTR. The next gen lead compound, we have CFTR protein being expressed. And if we zoom in on the right-hand side at 100x magnification, you can see that the orange of the CFTR protein is on the apical side of the cell, exactly where we would want it to see it being present. So in summary, there remains a significant unmet need for patients with cystic fibrosis who are not available to CFTR modulators or who gain little benefit from those. mRNA can be synthesized, packaged, and actually now is in a clinical study with MRT 5005. But beyond that, having advanced our platform, our technology, we now have a next generation molecule that we're bringing forward as we continue to investigate the biology and other properties of CFTR. So what I'd like to do in finishing is to acknowledge the patients and the investigators who have participated, continue to participate in the Restore CF clinical trial, the Cystic Fibrosis Foundation, the Translate Bio team, and thank you for watching this presentation.